Welcome. So, today we will still continue with that enzyme which is taking the help of molybdenum, and we are talking about some reaction which is the most fundamental reaction, that is the oxo-transfer reaction. And since molybdenum bearing enzymes are involved, so we call them as oxo-transfer molybdenum enzymes. So, last time we have seen about that DMSO reductase. Today we will first talk about sulfite oxidase. And these are very important group of enzymes, and they have several sources where from we get these sulfite oxidases. And the basic components, because while talking about this molybdenum center, we should not miss out what are the other components also present along with this particular center. So, the most important source is the cow's milk, mammalian livers, and in case of the sulfite oxidase, two molybdenum centers are present along with cytochrome B. So, other electron transfer agents which is cytochrome B. So, these cytochrome B centers basically they all we know as that they are iron centers, and those iron centers will be responsible for transferring electron to the molybdenum center, and this molybdenum center ultimately taking part in the oxo transfer to the sulfite group. And in the next one which we will talk about is xanthine oxidase. Having similar sources, as well as we can get it from some bacteria also, and here also two molybdenum centers are present and 4 iron sulfur clusters. Instead of cytochrome B, we have iron sulfur clusters and flavin adenine dinucleotide. So, these are nothing but they provide the right number of electrons at right potential to the molybdenum centers. So, these are basically the centers which will be responsible for transferring electron to the molybdenum center. So, when we talk about this sulfite oxidase, we will see that the individual centers what is present, this I also shown you last time, that after this conversion, that means the molybdenum is there and some groups, basically these red balls are your oxo groups. So, you have this diethylene type of disulfur coordination, then cysteine sulfur. So, this is also cysteine sulfur, and this particular oxo function. So, this oxo function and its transfer, it is all present in DMSO reductase, nitrate oxidase, aldehyde oxidase, and everywhere. These molybdenum centers, whether we can go for a dioxo system to a monoxo system and a non-oxo system. So, if we go from a dioxo system to a monoxo system, so that particular oxygen can be transferred very nicely to that sulfur center bearing the sulfite anion, not the DMSO center or dimethyl sulfide center. So, this is very important step, which is the last step in the metabolism of any sulfur compound, which can be your drug molecule, which can be any other sulfur bearing molecule where you want to oxidize the sulfur center, because sometimes we want to make in the synthetic chemistry also in the chemical synthesis what we find that if you have a sulfur bearing center, we want to make it as a sulfone or sulfoxide groups, and ultimately if we can break something, that means the carbon sulfur bond, if we want to cleave, if we can go for the carbon sulfur cleavage where you the sulfur is attached to the organic part. So, that is removing as the corresponding sulfur bearing anion. So, it can be sulfide, it can be your sulfate. And interestingly, the substrate hold, that means where you bind this sulfate group close to the molybdenum center, because you cannot have a very long range electron transfer, because all the iron bearing cytochrome B or ferridoxine molecules or the FAD type of molecules which are involved there. So, these two centers are close to each other, and several other residues are also involved for interacting with the substrate. So, this is the substrate binding pocket and this is your molybdenum site. So, close is just showing is the corresponding close proximity between these two centers, and sometime we will find that these molybdenum oxo centers, the suitable positioning of this particular oxo compared to the other one, because if you have, in most cases these are not in trans orientation, but when a cis diaxo molybdenum center, even in synthetic compounds, the model compounds will find they are oriented in the cis positions. So, when they are holding in a cis position, and if the molybdenum center is penta coordinated, it is close to a distorted square pyramidal geometry. So, you have a square base which is also, the molybdenum is sitting above this square base and one oxo is in the apical site, but this particular one is the basal site and this oxo is close to your sulfide site, and this oxo center is responsible for transfer this oxygen to your original sulfide group. So, when it is oxidized and formed a sulfate group also, so this particular one, when it is oxidized compounds, it can be excreted from the system. So, sulfide initially after formation, the sulfate formation, it also binds within the pocket of that particular oxidase enzyme. So, later on it is then removed from the system. Okay. So, these are the corresponding sites. We have already seen that how the different sites are involved. So, when we have this dioxocyte, so you have this diethylene part, so is the in diethiol. So, you have in function one double bond, carbon-carbon double bond, which is giving you the sufficient rigidity to the diethyl coordination, and the molybdenum, one side you have the cysteine sulfur and the two oxygen. And this particular one, though they are in cis orientation, but this angle is greater than 90 degree, this is above 100 degree sometimes. So, when it is going there, so when this formation of this dioxo species in +6 oxidation state is important, like that of our molybdenum acetyl acidulate, dioxo acetyl acidulate compounds. If you go for two of these two sites are coordinated and other two sites are also coordinated, so that will be hexa coordinated for the synthetic molecule, but here it is 5 coordinated only, and you see this, not this oxygen, but this oxygen is interacting with your sulfite anion. So, that is why these two centers are sitting within the enzyme pocket nearby. So, what is basically happening there, that this particular center, so cysteine sulfur is still attaching there. So, it is basically going from molybdenum +6 to molybdenum +4. So, this particular one, so this molybdenum all the time, even in the synthetic compounds will find that whenever you are handling something, that means the molybdenum mononuclear system. So, if you have a molybdenum nuclear system, then when it is attaching through these sulfide groups, it is basically forming, though this part is basically a anionic part, but you have something, that means you have this bridge between this molybdenum and sulfur through one of the oxygen, which is important. So, if you can go for some bridging unit, so in the intermediate state, this molybdenum can interact with some other species and it can go for some interaction where you can have a molybdenum sulfur dinuclear entity through bridging oxido group. So, when it is bridged, then this oxygen will be, there will be competition between this molybdenum center and sulfur center for this oxygen, and this molybdenum is in the reduced form. So, when this molybdenum is in the reduced form, it will have less affinity for oxo function. We all know that the oxido group or the oxido function is very useful for stabilizing the molybdenum center or any other metal center in the highest possible oxidation state. So, if you are able to reduce it, its affinity for oxo would also be less. At that particular point only, this molybdenum is providing this oxygen to the other anionic species. So, this is also very interesting to know that some other compound or some other molecules you can consider it also, that if you have the metal center you reduce it and it will just basically donate that oxygen to other substrate or the parent molecule. So, this oxotransfer reaction is very facile in that sense that if you have just reduce it and that oxygen can be transferred to the sulfite or any other substrate. So, this is the entity where the sulfate group is oxidized form and it is still bound to the enzyme function. So, this is the thing what we have seen in this particular one. So, molybdenum is there and you have the sulfate and already you have the sulfate, then you have the transfer, then oxo form is again formed over there, but still it is attaching to the system. So, either it will be attaching to the enzyme center or it can leave from the system, because we are not showing the protein envelope. What we are showing, we are concentrating our attention only on the molybdenum center and the sulfur center, but it is not like that. You have the huge envelope surrounding the molybdenum site as well as your sulfate site. So, when we focus our attention on the metal center for the catalytic point of view, we only focus our attention on the metal center, molybdenum and the sulfur center, but we are not focusing our attention on the entire protein envelope. So, protein envelope will be there. So, that will basically stabilize this formed anion. Formed anion is what? Formed anion is your sulfate, what is formed from the sulfide. So, there are some other positions also for the protein chain or the protein cluster, that this sulfate group then ultimately will leave the corresponding enzyme function and you will be leaving behind with that again a pentacoordinated molybdenum center, but your oxocyte will now be occupied by OH2, that means water. So, initially it is forming as a vacant site, but molybdenum will try to prefer or try to go for a pentacoordinated species in +4 oxidation state. So, it will immediately attract water from the system. So, it will attract water from the enzymatic system. So, then at this particular point again it will go for some amount of this particular oxidation. So, this molybdenum center, when it is attaching to oxo as well as water center, it can go for oxidation. So, one electron removal step can take place and you can generate something where you have this, instead of water it can bound to water as well as hydroxide group, because this OHN, that is why it is showing as OHN. So, N can be your 1 or N can be your 2. So, it can be water. It is very difficult to identify whether your water is bound to molybdenum center or hydroxide of function is bound to the center. So, what we are talking about for this oxidation reaction is we are settling between two oxidation state, which is +6 and +4. But the regeneration of the active state or regeneration of the +6 state is going through some intermediate where the molybdenum center is present in +5 oxidation state. So, how we can prove this particular form? That means, here you have only a monooxo species. So, this molybdenum center is basically a monooxo center and one water molecule is attached to that molybdenum center and it is in the molybdenum 5 oxidation state, which is a 4D1 system. So, you have one unpaired electron and that unpaired electron can be very nicely detected by EPR spectra. So, EPR spectrum will tell you that whether you are going from molybdenum 4 to molybdenum 6 via a transient or very weak lifetime for the corresponding molybdenum 5 center. So, that can be very easily detected when you pass from here to the other side. So, this spectroscopic technique, so for this particular one, that means it should be EPR active. So, in synthetic molecules also, whenever we make some molybdenum 5 compound and that can be immediately characterized, forget about its corresponding donor atoms or the ligand centers, you can consider it as that it is EPR active, that means it is in the molybdenum 5 oxidation state. And this particular loss in corresponding oxo group, that means the molybdenum dioxo species to molybdenum monoxo species, can also be examined by XRAFs, extended X-ray absorption film structure. So, that that immediately that is changing the corresponding oxidation state. We know the XRAFs technique is very useful to know the corresponding oxidation state as well as its immediate environment. So, when your immediate environment is changing from a dioxo form to a monoxo form, your corresponding feature for X-ray absorption fine structure is also be different for molybdenum 6 and molybdenum 4 species. So, XRAFS technique, so this corresponding characterization technique using XRAFS as well as EPR would be useful to identify any molybdenum site, because in most of the cases they will be settling between these two oxidation states, one is +6, another is +4, and at which particular form basically, that means it is transferring this oxygen to the sulfide group, and that particular species is responsible to know as this corresponding oxo transfer agent. So, what we will see that in some synthetic molecules, what we have seen earlier, that the most important example is MoO2 acetyl acetone. So, MoO2 acetyl acetone, so is a typical dioxo compound, so is a dioxo molybdenum compound. So, you can think of something that in all these model studies, because the model studies would be useful to know how good all these synthetic molecules also are useful to transfer this oxo group. So, model studies will tell us for this oxygen atom, the fate of this oxygen atom, whether it is going for addition to the molybdenum center or removal. At one case we are adding oxygen atom to the molybdenum center, in another case we are removing that one, and since we are talking something where you have this molybdenum and cis dioxide form, and basically we are talking about something that means oxo removal or oxo addition from the terminal sites. It is not by anything from the bridging side, that means at the intermediate what we are looking for the corresponding characterization of molybdenum site in the +5 oxidation state, that is not giving you anything where you have molybdenum oxo molybdenum thing is forming. That means the bridging oxygen is not involved there, the terminal sites are involved for transferring oxygen atom to any other substrate like your sulfite. So, like this acetyl acetone, you can take some good example for another ligand is dithiocarbamate. Dithiocarbamate, you can make it from diethylamine. So, what we are looking for is that you, instead of not getting anything related to simple that diethylene molecule, but a sulfur bidentate ligand is useful. So, at terminal sites when they are forming, that means we are looking for some oxidation state of +6 and +4, and at intermediate we are getting +5 oxidation state. So, if we get that compound, the DTC, the dithiocarbamate through these two sulfur groups can attach to your molybdenum. So, is a dioxo molybdenum in +6 oxidation state, and what we are looking for? We are looking for something where you can get a corresponding oxo transfer to any substrate. So, one well known oxo transfer species is triphenyl phosphine, and most of the cases we take the we can abstract this oxygen from the molybdenum center. So, this immediately can give rise to a compound where you can make the corresponding molybdenum 4 compound, which is monooxo molybdenum, and O PPH3. So, what you want to know that you can have this sufficient stability for the molybdenum in the +6 oxidation state as well as the molybdenum in the +4 oxidation state. So, but we all know that in case of say copper 2 and copper 0 and all these. So, if you have these two oxidation state, one is +6, another is +4, and interestingly from the terminal sides. So, we can expect something which we can call it as a conproportionation. So, whether we can get some conproportionation reaction over here or not, so conproportionation reaction, that means if you have these two, so when they interact to each other, you expect something which is molybdenum a dimeric form. So, molybdenum 5,5 dimer. So, how these two can interact? Is molybdenum oxo molybdenum dimer. That means, we are looking for in the enzymatic cycle the intermediate. What is that intermediate? So, if we get that, this is the corresponding oxo form, this is the corresponding oxo, and two DTC ligands are still attached to it. This is 5 and this is 5. So, from the techniques, two techniques what I just told you, that one is the European technique and another is the EXAPS technique, and these two techniques are sufficient to tell you whether a monomeric molybdenum 5 is in the active site or as the intermediate, or a dimeric molybdenum oxo bridged molybdenum 5,5 compound is the corresponding species which is there in the active site. But all these spectroscopic measurements will tell us that no dimer is formed. At the active site. So, that particular dimer formation is prevented, and in this particular case, even if we just talk about the simple dithiocarbamate. So, if R is your ethyl, we get diethyl dithiocarbamate. So, at the terminal point, that means this particular part, that means if you give for this, any of these oxo center is responsible for the formation of this dimeric one. But in the enzymatic reaction, you have the typical envelope, the protein envelope is there, and another protein envelope is surrounding this molybdenum site. So, the steric bulk of these groups which are responsible to bound to the molybdenum center will prevent the corresponding dimerization reaction. So, any synthetic molecule which bears a bidentate ligand of large size. So, large multidentate ligand, large multidentate ligand having some sufficient amount of steric bulk such that your one of the species which is forming over there, and after reduction is giving you the molybdenum 4. These two cannot interact. This interaction between these two form can be prevented. So, the multidentate ligand large multidentate of steric bulk will prevent dimerization. So, that way we can prove that only the mononuclear system is present and mononuclear species is responsible for the corresponding oxo transfer to the sulfide group. So, once this is formed, that means the sulfide is therefore attacking the sulfide oxidase and your sulfate is formed, and you are transferring nicely this oxo group to the inorganic anionic form, that means the sulfide group. And this we all summarize here, that the active site of sulfide contains the molybdoterine cofactor. In last class we have discussed that what is that your molybdoterine cofactor, and this particular sulfur coordination in around a double bond or in backbone is basically stabilizing the molybdenum in its highest possible oxidation state, that means the +6, and the enzyme in the oxidized form is coordinated to the cysteine thiolate, and another interesting group we call it from the molybdoterine unit is your diethylene group. So, what is that diethylene group is very important to know, is a huge group of molecules, and people have started knowing all these things when this particular molecule is known, that what are these diethylene groups, because it has other immediate applications. So, diethylene ligands is a separate class of molecules which can bound to molybdenum, not only molybdenum, to nickel, to any other groups, and they give rise to very useful materials. So, like that of your acetyl acetone or any other class of ligand system, diethylene also form a distinct class of ligand system. So, this diethylene group of ligands are useful for the coordination of molybdenum, and you already know that you have the 2 terminal oxygen atoms, and the manipulation over those 2 oxo groups are basically giving rise to corresponding oxygen transfer to the sulfide function. So, is transferred to sulfite and sulfate is formed, and water is then displaces the sulfate, and the removal of 2 protons and 2 electrons returns the active sites to its original state. So, how we get back the cycling, and all this, every time I am talking about this, that in all these cases your dioxygen is not taking part for this oxo transfer reaction, you have to have use of the corresponding water molecule. So, it is a water mediated reaction, that is why this reaction is also very interesting, that this particular one is very useful, and we are able to go for some oxygen atom transfer, and that oxygen is originated from typical water molecule. So, that is why it is very interesting to know that any coordination chemistry related to the metal center, which is initially coordinated to the water molecule, then it is coordinated to the hydroxide function, and ultimately coordinated to the oxido function. So, how easily we can generate this oxido function from the water molecule, that this catalytic cycle for this class of enzymes also show us that you generate the oxo function obviously by taking the help of molybdenum center, and that oxo is very much useful to transfer to any other organic substrate. We do not require any such other reagents like this hydrogen peroxide or MCPB and all these things to go for this corresponding oxo transfer, but your source is simple water molecule. So, these are basically what we have seen for these the different steps for the catalytic cycles, and ultimately it is the water molecule which is taking part for oxo transfer reaction. Okay. So, this group of molecules, when coming to us, we will see that whether this particular oxo group like that of your dimethyl sulfoxide reductase or sulfide reductase. So, these are some kind of organic solvent type of molecule or typical anions of sulfide base type molecule. This particular group, which is already we know that this diethylene part, so dithioketone part at the dithylate part, because basically so this in dithylate part, sorry, this is dithioketone, it is changed. This 1,2-dithioketone is this one. This is 1,2-dithioketone, and in 1,2-dithylate is the left one. So, whenever we bind this particular one and molybdenum is in this particular form, and your sulfur is negative and this sulfur is also negative, and you have the molybdenum in the +6 oxidation state. So, why we are talking that these are very interesting molecules, so they give rise to some metal diethiolenes. So, these metal diethiolenes have tremendous applications also in some other areas, and due to this delocalization exists in multiple oxidation states, because is sometime we talk about like some other ligand system, whether your ligand is in the oxidized form or the metal center is in the oxidized form. Like in the case of copper, we have seen in the case of galactose oxidase the tyrosyl radical formation. So, any organic part, so this diethylene part also you should be very much careful whether the metal center is bound in dithioketone form, this is the dithioketone form, so dithioketone form, or in 1,2 diethylate form. So, this basically, this knowledge basically the identification of molybdenum in all these molybdenum enzymes, oxotransfer molybdenum enzymes has generated tremendous interest to the scientist or some synthetic chemist. That is why this is a very old paper series in 1962 in a Jack's paper. The reaction, you can see this paper also is a very interesting one. It's a diphenyl acetylene with nickel sulfides. So, diphenyl acetylene is there and we are reacting with nickel sulfide. So, this diphenyl acetylene, this will be pH. So, diphenyl acetylene, so you have the direct sulfur attachment to the triple bond of diphenyl acetylene. So, that gives rise to the generation of this sort of ligand system, and which is already, so you have a modified ligand system based on nickel sulfides, and you have the in 1,2 diethylate group which is attached to the nickel center. So, these diethylene molecules are very interesting and they give rise to different interesting molecules, to vanadium, to molybdenum, to copper and all these. So, depending upon the presence of this metal center and the corresponding delocalization, because these are very small molecule species, you can burn this at high temperature in the oven and you can get a different type of corresponding metal sulfides. What you are reacting is a one type of nickel sulfide, then if you can burn it, you get a different type of metal sulfide which have some tremendous materials application. Then next we will just go for the corresponding system which is our xanthine. So, xanthine molecules what we get is the corresponding form what we get from guanine. So, xanthine we can make from guanine through the use of guanine deaminase. So, not only xanthine, there are several other groups of molecules which are also interesting. And you can have 3 substitutions R1, R2 and R3. These can be your hydrogen also. So, related to xanthine, so xanthine molecule will come there. So, related to xanthine molecule, some other type of molecules, all these. So, they are involved in related to our purine metabolism, purine and pyrimidine. So, purine and pyrimidine very important molecules. So, these molecules when we go for the corresponding metabolism in our body also. So, purine metabolism and there are several other sources also present that is involved for the corresponding assimilation of these from the natural sources. So, one such we can just simply know that when R1 is equal to R2 is equal to R3, that means when all the substitutions are methyl, we get caffeine. So, your source definitely would be coffee, tea, cola, etc. So, we get all these from coffee, tea, cola, other things. So, simple manipulation of the substitutions R1, R2, R3, we just basic skeleton remains same like that of your guanine, that of your xanthine, all are same. So, either they are all pyrimidine bases. So, now if we just simply make R1 as H and other two R2 and R3 are methyl. That means, one of them you make as the corresponding protonated form, which is also has some medicinal value, which is theobromine, which is available from chocolate. So, and then the other one, that means you change R1 and R2 what we had as methyl, but you make R3 as H. So, one case R1 is H, in another case R3 is H, which is theophylline, which has medicinal value also, which is also present in tea. So, if we want to metabolize these, that means how we are metabolizing the sulfide group to sulfate. So, basically any other sulfur bearing molecule can be oxidized to sulfite and then to sulfate. So, we need some oxidation reaction. So, Zanthinoxidase is that molecule this Zanthinoxidase molecule will be responsible for converting. So, if we will have another related molecule which will be getting from the purine bases. So, xanthine in that particular case will be converted to uric acid. That is why we always talk about uric acid, and the blood content of uric acid is also very important to know. And this particular one is catalyzing by molybdenum. So, you have molybdenum 6 and molybdenum 4. So, therefore, your Zanthinoxidase has molybdenum center, and which will be responsible for another type of oxotransfer reaction. So, this can also be considered again as oxotransfer reagent. So, this very good oxotransfer reagent can be responsible for oxidation of xanthine to uric acid. So, this is a big group of molecules. So, Zanthinoxidase is a form of xanthine oxidoreductase. So far, we are talking about the molybdenum oxidoreductases, that means the molybdenum bearing oxidoreductase, but this is a very specific one. We all know now that the corresponding coordination environment and what are the groups which are directly attached to your xanthine center, Zanthinoxidase center. So, depending upon that, you get a corresponding oxidoreductase which is known as xanthine oxidoreductase. So, this particular one can be responsible for generate the reactive oxygen species ROS it can generate, and it can catalyze the oxidation of hypoxanthine to xanthine and can further catalyze the oxidation of xanthine to uric acid. Not only the simplest possible transformation of xanthine to uric acid it is catalyzing, but also it can catalyze the corresponding formation of xanthine also from the hypoxanthine molecules. So, what we get in our body also, in some other all living systems, we all have the purine bases related to your nucleosides and nucleotides. So, all these purine bases we can have, and these purine bases, if they are getting oxidized, particularly these two, the adenine and guanine. So, adenine and guanine can be giving rise to some xanthine molecules, and those xanthine molecules can ultimately could be converted to uric acid. So, how we get that is also another biosynthetic pathway or biosynthetic process where you get xanthine from guanine. So, your source is the guanine molecule, and that guanine molecule is giving rise to xanthine molecule. So, structural correlation between all these groups, all these bases, all these purine bases, we should know little bit that what type of transformation is taking place when you move from guanine to xanthine. So, if it is simply going for some removal of NH2 function, we call it that is the guanine deaminase. So, by looking at the corresponding reaction, if we just simply look at the corresponding reaction, that one extra NH2 group is going away, and that particular group either it is in the diamine form or it can be attached to something where you have the oxo center there. So, molybdenum we all know now that it is a very good reagent, very good species like that of your sulfide to convert to a sulfate species. it can go for corresponding hydroxylation on the carbon center. That means, the carbon based hydroxylation is also possible over there, and that carbon based hydroxylation reaction is also giving rise to corresponding uric acid. So, this particular one, so in this particular case, how you get xanthine to guanine is the guanine deaminase reaction, and but in this particular case we are not talking about guanine deaminase, but we will be just concentrating our attention on how your xanthine molecule is getting converted to your uric acid molecule. So, the basic reactions, these are the several reactions what we can have. That means, you have the xanthine, and that xanthine you have certain number of oxygen groups attached to this particular center, and which is converted to your uric acid. So, basically we are going for some oxygen atom transfer to the substrate system. So, what is our xanthine? So, xanthine molecule is nothing but related to this particular caffeine, theobromine or theophylline type of molecule. So, is again something derived from your purine base, double bond, oh no, is one double bond is here also, this is aromatic. So, you have this is hydrogen, R3 is hydrogen, and this is OH. So, you are using Zanthinoxidase, and that Zanthinoxidase in all cases we are discussing so far is the oxygen atom what is coming over there is derived from your water molecule. So, you are reacting it with water. So, this OH in one form you can write it double bond and NH, this is all same. So, you can write it as a double bond O and NH function. So, this basically going for its corresponding uric acid. This uric acid is formed plus hydrogen peroxide. So, in some cases also you have another site, this particular site and these two particular site, those can also be hydroxylated. So, basically what we are looking for? We are looking for something where we have all know that aromatic hydroxylation reaction. So, benzene to phenol or phenol to catechol type of conversion. Hydroxylation reaction, true hydroxylation reaction such that you get a CH function to a COH function. So, this particular one, so this once this uric acid is formed and this uric acid formation to us to anyone's body is forming the corresponding state which is related to your gout formation, because the uric acid crystals are forming there and that is giving you to severe pain within the bone joints. So, this particular one, that means that typically now in this particular case of molecules we can transfer this oxygen, we can go for the typical hydroxylation reactions what we see in case of copper mediated reactions or any other cases. So, you have xanthine, so therefore, you straight away we write it xanthine plus water plus oxygen giving you uric acid and hydrogen peroxide, and the formation of xanthine is also in the similar fashion, it is catalyzing by water and oxygen and forming hydrogen peroxide over there. So, xanthine oxidase can also act on certain other purines. So, this is interesting. So, any kind of purine like substrate if we can have. So, that gives us some important clue that what type of molecule you are taking over there, and that is responsible for the corresponding hydroxylation reaction or the pterines and the aldehydes. So, xanthinoxidase is effective for some kind of other purines, pterines and aldehyde molecules. So, if you have a high level of xanthinoxidase in your body, so if the substrate molecules like xanthines are available, it will immediately convert those xanthines to uric acid. But if your substrate concentration is not that much, it will just act on the other molecules available. If the aldehyde function is available, it can be oxidized that aldehyde to acids and other oxidized form. So, some selectivity is also there. It efficiently converts 1-methyl-xanthine. So, 1-methyl-xanthine is already we have seen the caffeine is the all the 3 positions are the methyl functions. So, one of them is the 1-methyl-xanthine, which is the metabolite of caffeine molecule. So, metabolite of caffeine molecule is there, and that can immediately convert it to 1-methyl uric acid. So, you see that if you have caffeine in your body also, so is the body source what you are increasing by all these type of molecules, not only that you have the purine base from ATP and all other things. So, that particular one, that means if you get this through some other sources, particularly the food sources, so that will also be converted to some other molecule which is related to your corresponding increase in concentration of uric acid. It is only a substituted version of uric acid. So, you will be able to form methyl uric acid, but it is not active on 3-methyl-xanthine. So, it is only active on 1-methyl-xanthine. So, there is some selectivity definitely that one particular substrate in the biological system, the selectivity is much more, which we cannot get in the laboratory. And in some cases, we will find that when you have the corresponding reaction on some alkane type of thing, that is the methane hydroxylation to methanol, that it can produce in some cases, it can also produce some superoxide anions also. So, basically in these particular cases, if you have the dioxygen available and the dioxygen is taking up that electron and you are getting some superoxide anion, and that superoxide is involved for further transformations. Okay. So, now we just simply compare, because we know now what the caffeine, the theobromine and theophylline. So, what is that your hypoxanthine? So, now you can corresponding relationship with the guanine molecule. You know the guanine structure, you should know that typical guanine structure. So, if you have this NH2 function and one more NH2 function, so you just simply remove the NH2 functions. So, NH2 functions are removed, it is not remaining as a corresponding CH or CH2 function, but some oxidation reaction is also taking from there. So, these 2-3 molecules basically we can correlate that in this particular case the hypoxanthine has one oxygen atom. That means you have transferred one oxygen atom to the parent molecule related to your purine base, that means the guanine, where you have extra NH2 functions also. If you can go for 2 such points, that means this carbon is going for the hydroxylation reaction. If you go for the second carbon, you get a molecule which bears 2 oxygen atoms, which is your xanthine, and if it goes for the third one, you get the uric acid. So, you just one particular substrate. So, the enzymatic reactions are so important that one after another you can insert 1 oxygen atom, you can insert 2 oxygen atoms, and you can insert 3 oxygen atoms. So, ultimately the corresponding one, that means the crystallization of uric acid, because their solubility and the corresponding anionic form dependence on the pH, all these things will depend on how much uric acid is there in your body or in your blood, and that will also be related to your gout formation or the diseased condition depending upon your uric acid formation. So, molybdenum will definitely be important there, and the molybdenum is responsible for giving rise to this particular reaction where you get the corresponding hydroxylation reaction. So, how we get, that means there are something, that means you have the entire protein structure, and we are writing something that you have iron also and you have the FAD molecule also. So, the entire protein structure, which is definitely a complicated one because it has a very huge molecular weight of 270 kilodalton. So, it has 2 FAD molecule, 2 molybdenum center and 8 iron atoms per enzymatic unit. So, we are talking about one molybdenum center, and these molybdenum centers, which are your molybdoprotein cofactors and active citrate enzymes, and it is accepting the electron from your iron centers, and these iron centers will provide you the right number of electrons at right potential, the E0 values, which are important. At what E0 value you will be able to reduce the molybdenum center. So, that is why we are talking about the molybdenum centers with different environment. So, if you move from one environment to the other, particularly that typical one which we are seeing, the sulfur environment, the diethylene environment. So, is this diethyl environment is corresponding diethylene environment depending upon its electronic structure. This diethylene environment is completely different compared to your cysteine environment. That means, it cannot be equivalent to 2 cysteine residues related to your molybdenum center. So, if you are able to make synthetic molecules also bearing those sulfur groups from this cysteine sulfur or any other sulfur from that corresponding thiophenol and all this, will find that corresponding double bond, the in diethyl unit. So, that electronic structure or the electronic environment related to that in unit of the diethyl is different. That is why if you just measure the corresponding E0 value, the diethylene ligands will give you some other E0 values. So, it is giving most of the time is a very low potential for the corresponding center oxidation. That means, its corresponding charge density or the electron density on the diethylene is much more compared to its other corresponding diethyl ligands. So, iron sulfur clusters are responsible. So, ferredoxin type iron sulfur clusters are responsible for electron transfer. So, obviously, this particular iron center at particular E0 value is used for transferring the electron to the molybdenum center, and molybdenum center will also be known at what potential it can be reduced depending upon its corresponding environment, whether you have a diethylene environment or any other environment. So, the active site of the xanthinoxidase is again similar to that of your sulfide oxidase, and you transfer this particular one from molybdenum to xanthine, again is a typical that oxotransfer reaction that of your sulfide, and you just regenerate back the corresponding active molybdenum center, which is generated from there with the addition of water molecule. And other known molybdenum containing oxidoreductases, as we are talking all the time, the same thing. That means, it is the water molecule which is providing that corresponding oxygen to the molybdenum oxo center. So, any molybdenum oxo compound, if we can consider whether it is a monooxo molybdenum compound or dioxo molybdenum compound, if we are able to reduce that center, which is important, that whether you are able to reduce that particular, so that means use of that thing in presence of some reducing agent. So, we will be using some reducing agent, and we reduce the molybdenum center to either to molybdenum 5 or molybdenum 4, then you can take up that oxygen from the molybdenum center for some suitable conversion, whether it is a conversion from the sulfide or it is a conversion on the xanthine molecule to your uric acid. So, this molecule, so little bit has some relationship with that of your structure of the xanthine molecule. So, if we get the corresponding structure of that xanthine molecule, what we have seen that that corresponding xanthine molecules, that means the purine base. So, if you have the purine base, so that purine base we can have, and that purine base basically is our substrate molecule. And we are going for the corresponding hydroxylation reaction. This purine molecule will have CH bond, and this will be converted to COH, and the corresponding specificity of the enzymatic reaction will immediately tells us that one particular position, this nitrogen position. So, this particular carbon, this carbon is your center where you attack by this corresponding molybdenum oxo center. So, this oxygen can be transferred to this carbon center, which will be utilized for this corresponding hydroxylation reaction. So, uric acid has that particular structure also, only thing that you have the corresponding hydroxy function on this carbon. So, that corresponding remedy for this corresponding accumulation of uric acid from purine metabolism, how you stop the accumulation of uric acid. So, you have to find out something where you can have low concentration of uric acid, the generation of uric acid should be less there. So, if you have something, that means xanthinoxidase also can produce superoxide, that we have seen when it is attacking to your dioxygen molecule. So, this superoxide, we all know that the superoxide is also a very deadly system or deadly molecule, and which can damage your serum and lungs, and activities increase when we have the influenza A infection. So, the corresponding reactivity pattern for xanthinoxidase, where you have the corresponding reactivity for the xanthinoxidase, the first thing what you should know, that the amount of xanthinoxidase in our system, any living organism, any living system, how much xanthinoxidase you can have, which can oxidize your that purine molecules to your corresponding oxidized form, and at the same time, because it is producing hydrogen peroxide as well as it is activating your dioxygen molecule to superoxide, because it is also related to some other diseases, that means that is corresponding marker for the concentration of xanthinoxidase can also tell us that how much activity is for generating your superoxide molecules. So, during this particular cases, that means during this disease condition, your xanthinoxidase can be released into the blood. So, blood assay for xanthinoxidase. So, there are two typical things that we will discuss in our next class, that if we can have the blood assay for xanthinoxidase, that means what we are measuring your level of xanthinoxidase in your body. So, depending upon the concentration of the available xanthinoxidase in your body and depending upon the purine concentration, that means the guanine concentration, you can tell that how much uric acid will be produced by the concentration of the available xanthinoxidase. But in some cases, if we want to stop or if we want to inhibit the corresponding reactivity of this corresponding reaction of this xanthinoxidase for the uric acid pathway, so this we call it as some inhibitor design. So, all these medicinal things, all the medicinal chemistry basically related to some designing of inhibitors, and inhibitor designing is very simple, that we will see in the next class, that one such molecule is allopurinol, which can be used for the treatment of gout, which is nothing but this particular structure will have very much similarity to that of your xanthine molecule, only thing that it cannot be hydroxylated. So, you have the substrate where this function, that means this CH2 function is, this particular backbone is missing. If you do not have this particular carbon, that means if you just bring this nitrogen over here, that means you carry this carbon over here and make these two nitrogens. So, you see the selectivity is such an interesting one, that means once you make this carbon as nitrogen, it cannot react with the molybdenum oxo form to get it hydroxylated by N hydroxylation reaction. So, N hydroxylation reaction is not facile there and it will not form there to give you some N hydroxylated product. We will just looking for uric acid formation, the C hydroxylation reaction. So, C hydroxylation reaction is important. So, anything, any molecule related to your xanthine which can stop or which can inhibit the reaction of xanthine oxidase will serve your purpose and will behave as an inhibitor and your corresponding medicine for the treatment of gout. Okay. Thank you.